So now we're going to do something a little bit different. The next two talks, we're going to drill down in a more slightly, well, I shouldn't say more experimental way, but in a mouse kind of way, um, and and try to look at some underlying mechanisms from that point of view. And our first speaker actually does need an introduction because she's brand new here at Einstein. Renata Batista Brito is an assistant professor in neuroscience, recruited here only in April or so of this year. But her lab is all set up and she's ready to go. And she's going to talk to us about inhibitory systems in mouse models related to autism.
Well, first of all, thank you, the organizers, to invite me to be here. As it was alluded, we are going to change gears now a little bit, and what I'm interested into is to see how basically you can use the mouse system as a way of sort of dissecting apart some of what it can be endophenotypes of neurological disorders. So, psychiatry disorders such as autism, schizophrenia, and others are very complex and integrate the whole brain, and therefore they cannot really be models of autism in mouse per se, because it's a very human disease. Now, and when we think about the brain, we think about this homogeneous system that is composed by lots of structures, but that sort of acts together. But that is true, but it is also true that the brain is constituted by a variety of different networks and different substructures that are composed by a variety of different types of neurons. And these different neurons might be in these different systems might be differentially affected in different aspects of disease. So in my lab, we take the advantage of the fact that in mouse we can do genetics, and we can certainly manipulate specific aspects of the network and specific areas of the brain. And we can combine that with manipulations that allows us to also record from cells and manipulate cells of a specific type. And so we can probe the activity of the brain in a mouse that is engaged in a task and that is freely moving at its own will, and assess how different mutations that have been linked to disease might be affected in certain circuits. And so what I'm interested in actually how do different subsets of inhibitory neurons might be involved in neurological disorders such as autism as well as schizophrenia. And this is a very simplistic view of the cortex, of the visual cortex, but could be other cortex, in which the vast majority of cells of neurons, as you know, are excitatory pyramidal neurons. But if this was all the neurons we had, we would be constantly seizing. So of course they are assisted by a variety of inhibitory neurons or interneurons that project locally and that are very flexible in the way that they mediate the response of excitatory neurons. They can be fast spiking cells that provide really reliable fast inhibition. They might be VIP interneurons that are mainly target other inhibitory neurons. And this allows the cognitive capacity and the sort of the computational capacity of the network to really exponentially increase to lead to different responses depending on the circumstances, and it's being associated with not only pathology but also brain flexibility. So today I'm going to tell you a story about neuron type that is a vast minority, is a small minority in the brain, is the VIP expressing interneurons. They are less than 1% of the neurons that you have in the cortex, and so they have been sort of underlooked over many years. And there's not really much known about those in the context of disease. However, I postulated in my postdoc that this would be an interesting interneuron type to look into, because even though they are only 1%, they receive a bottom-up input, such as thalamic input. They also receive long-range input and therefore top-down input, and they are targeted of neuromodulatory inputs such as acetylcholine and serotonin. And all of these are sort of characteristic markers that might be affected in autism and in schizophrenia. So I thought it would be interesting to ask, what is the involvement or the possible involvement of this cell type in disease? Also, there was some indications that these cells might be depleted in some patients. So that was the motivation. So to get at those cells, I decided to knock down specifically one gene called ERBB4. There is a gene, I'm not going to get into the details for time's sake, but it's a gene that has been linked to both autism and schizophrenia, and specifically target in VIP interneurons, even though it's expressed in other cell types. So when we do that, and then we can ask, what happens to the VIP interneurons, these inhibitory neurons itself, when we record them in a mouse that is on a wheel and can freely move. Now, in my experiments, I'm going to show you either calcium imaging that we are going to use as a proxy for firing activity of cells, or extracellular recordings. In this case, it's an experiment with calcium imaging. So basically, this is the calcium. So the cells express a calcium indicator, VIP cells, and these are control cells in a mouse. And you can see that VIP interneurons are fire or become active when the mouse starts moving. So this is locomotion onset. You can see most of them are not responding, so are silent. And as the mouse starts moving, they become very active. That was previously known. What was surprising is that in the mutants, this regulation is totally gone. So basically, these cells that are highly induced by locomotion no longer can tell. The network can no longer say whether the mouse is moving or not. So then we wanted to go farther and ask what happens to the other cells in the cortex. So what happens to the excitatory neurons, to the pyramidal cells? And what happens to the relationship between neurons firing with each other? So we recorded in simultaneous local field potentials, which gives you the sum synaptic activity of the local network, sort of a narrow EEG, and single units of many, many cells at the same time, in the control and in the mutant. And then we ask a variety of questions, and I'm going to show you some of the data. So the one thing that we notice immediately is that, in the controls, always here and for the rest of the presentation represented in black, individual cells are firing in line with the ongoing rhythms. For instance, here we have a rhythm we are looking at the spike trigger LFP averages that shows you how the cell is entrained to the network. And you can say it's nicely entrained in the control. But in the mutant, that entrainment is totally gone. It was true at the 1 to 6 frequency and at the gamma frequency, 40 to 60 hertz. And it's like if they are, snowflakes, not really following the undergoing activity. We also looked at synchrony, how cells fire together, which, if they receive the same synaptic input, they should fire together. And you can see that synchrony is also totally gone. So then we ask what happened to the firing properties of these neurons. So here you have an example of a control and a mutant, and here the population analysis. And you can see that the firing activity of excitatory neurons, the pyramidal neurons, in the mutant mice is massively increased, about 6-fold increase. Well, then we ask, okay, so if the cells are not entrained to the network and they are firing much more, what happens to the capacity of the network to respond differentially when an animal is in different behavior states, this capacity of adapting to different circumstances. And here there's an example of one cell, one excitatory neuron, pyramidal neuron, in a control mouse, and you can see that usually they increase their firing activity to locomotion onset. This is the population analysis. And in the mutant, that modulation was totally gone. Again, like the network, from the perspective of the majority of cells, excitatory neurons, could not tell whether the animal was walking or sitting. And this is the quantification. So we are doing all these experiments in the visual cortex, and the reason is because then we can ask how does visually evoked activity change in these animals? Is there any deficits, kind of speaking to very much of what John just told us about? So we did similar experiments, but now the mouse, instead of being, not observing anything in particular, is seeing a drifting grading that is presented within their receptor field. And here you can see the response. So when you give this drifting grading to the mouse, which is a bar just like going into the screen, you can see that there's a visual response of this is the population of cells, and then there's adaptation. Here is the quantification of the signal to noise ratio of this response. And in the mutant, there's basically massively reduced again. So these mice have reduced visual processing or visual responses. And now, so I told you that I did this mutation in which I remove ERBB4 just from VIP cells, and I'm assessing what happens to the network. But when I did this mutation, I didn't give you the details, but I'm removing it for every neuron in the brain that expresses ERBB4. And I was doing this around P7, which is, 1 week before eye opening, and it's just 1 week after birth. And so I was wondering, what is this? Is this a fact that we are seeing something that is local? So I'm recording the visual cortex. Is this something that is happening in the visual cortex, or is being inherited by another structure and is basically illustrating something that is happening in the thalamus or somewhere else? So to address the question, we did a rescue experiment in which we put back ERBB4 specifically in VIP interneurons after we had removed it. And we did this at different ages. And so the one that I'm going to show is that we put back ERBB4 1 week after we removed it. So we removed it P7, we reintroduced it around P15, and then we ask only in the visual cortex. So now we are only rescue the part that we are actually analyzing, and then we ask what happens to the animals. So here's some of the measures. So you can see that the firing rate is totally rescued. So you have here the control, which actually it's, in this case, in the control and the rescue looks exactly the same. The mutant would be like totally different. Here you can see the state modulation. Again, control and mutants look the same, which would look very different. Controls and rescues look the same, which would look very different from the mutants. So now all of a sudden, these animals can respond to locomotion onset, or you can measure this response in the cortex. And visual responses were totally rescued. And that was quite impressive, I felt, that you can remove it, you can put it back just in the visual cortex, and you can rescue all these phenotypes. Now, the reason why I did this manipulation around P7 is because when there's a lot of the synaptic first interactions ongoing, there's a lot of plasticity, but the circuit is pretty well set. The cells have migrated, they have reached their targets, and etc. And then I'm very interested in my own lab to understand, what are the development trajectories of when these processes happen? What happens when you remove one element of the circuit, when synaptic transmission just starts, or when visual input just comes in at eye opening, and etc. So I've actually these rescue experiments at different ages, and when you rescue in adults, you could no longer rescue at all. So there's a critical period, there's a window of opportunity after there's already the onset of the phenotype, but before it's too late. That is kind of the sweet spot to manipulate this. And just to clarify, so the manipulation, the removal of the gene from VIP interneurons was at P7, and we started to see effects around P15, which is in this case when we corrected. So then we wanted to go all the way from a mutation in a gene, in a specific cell type, in a specific circuit, to a behavior that is involved with that circuit. So we trained our mice, controls and mutants, to a go, no-go visual detection task. It's very simple. The mice are water deprived. We train them so they are very motivated to learn this task. We train them to identify, a stimuli, which is similar to the ones that we gave before, these bars. And if they see it and there's a stimuli and they leak, they get a water reward. If it is a no-go, they get a timeout. And they learn this task fairly well. And first we train them in 100% contrast, so it's very salient stimuli. And as time goes by, we train them to detect like lower and lower contrast to a threshold that is really low. And for those who think that mice don't see, I get this question all the time. So the threshold was about 0.3% of contrast, which was actually as good as I did. So it was about the same as humans. The big difference is that for the mice, it took a longer time to learn than for me. But the threshold was about the same, so I was just better at it earlier. So here you can see the contrast curve, the performance curve. So here is the hit rate, how much time they get it right, of a mouse that is a control, early, which means like in the first days of training, first 2 days, versus late, which is like 2 weeks after it's been trained in this paradigm. So this is like a novice mouse, and this is an expert mouse. And you can see that they are always good, even when they start at really high contrast. At really high contrast. But in the beginning, they are really bad at low contrast, and they become much better, right, with the threshold being, around here. So in the mutants, these never happen. So the mice, basically, early versus late, are meaning a novice versus expert mice always performed at the same level. So this suggests that they have a visual detection threshold that is lower, or that they don't learn the task. We are now doing experiments to tease those two apart. And so here is the quantification, seeing that there's an improvement in performance in controls that is non-existent in mutants. And so here I'm just going to summarize with what I told you and some things I did not tell you, just to kind of bring the story together. So we were interested in seeing how these interneurons, 1% of cells, might be involved in disrupted off circuits associated with autism and schizophrenia. We removed the gene ERBB4 from VIP interneurons. When we did that, actually, we reduced glutamatergic input into these VIP interneurons. I didn't show that data because of time's sake. That led to changes in the pattern of activity of the VIP interneurons, where they usually are activated by locomotor signals that no longer could sense. In its own hand, it changed the connectivity between VIP interneurons and somatostatin interneurons, or at least their synaptic interactions. This led to massive consequences at the level of pyramidal neurons. And in terms of those, their firing rate increased. They were no longer synchronous with each other and no longer in tune with the network. As a consequence, we had massive changes or incapacity of cortical adaptation to changes in transition state. We have reduced visual responses and impaired or reduced performance to a visual task. And so this work was done during my postdoc at Yale in the lab of Jess Cardin, that I want to thank along with the rest of the lab. And this was my funding agencies during my postdoc. And now I'm just starting, I've been here for a couple of months at Einstein. This is my team. And we are following up questions that are related with autism and schizophrenia, and that basically very much wants to address what happens in these first couple of weeks in a mouse life that would correspond to maybe the first 2 to 3 years in a human, that makes the system so permissive or to these alterations. Is like changes in the receptor field, in the maps, what it is, and how can we possibly correct it by basically going from an engineer perspective and dissecting each component apart in the mouse. Thank you.
So we have time for a question or two. Renata, I think it was Ramon y Cajal who argued that the complexity of intrinsic neurons in the cerebral cortex in humans was greater than that of lesser mammals.
Yeah.
Can you say anything about the VIP positive cells in human cortex?
Yes, sure. So, that idea has been there since Ramon, and actually, I think over the last couple of months has been challenged. And it's been a bit like the idea of the genome, where we thought that, as we were going from the human to the mouse, it would be a massive change in the genome, and, it would be so much more complex, and everybody became very shocked. And it turns out that 99% is the same, right? So now there's been a huge effort, and I'm involved in some of those efforts, in actually compare cell types by using mainly single-cell sequencing, but in combination with some electrophysiology and other paradigms, and compare all cell types across humans and mouse, and mainly in the visual cortex, because most of these efforts have been pioneered by the Allen Brain Institute in the visual cortex. And they have exactly the same numbers, except for one cell type. There's apparently one cell type that exists in humans that does not exist in mouse. It's an excitatory neuron. But there's about 63 types that they came up with when it's subdivided in more finer tunes of inhibitory neurons, about 73 or 74, depending what it is in humans. So it appears that the big difference is actually the number, right? It's like maybe a scaling capacity rather than a type capacity, which was exactly what was in the genome as well. But so in terms what we know about them about these cells in humans, they are present, they have similar morphology, they have similar gene expression, they have similar firing properties. Everything that I've seen thus far, mainly coming from the island again, is that they are basically the same.
Other questions?
So, with this particular animal model, what behavioral aspect of autism do you think you could approach best, right?
Yeah.
Through this interneuron.
So in this manipulation, we did a few. I did a few behaviors that were more generic behaviors in the generic deletion of VIP interneurons, such as anxiety, social interaction, nest building, marble bearing, and many of those, we can go into the details, but many of those were altered in ways that could be seen as within the spectrum of autism. In the one that I'm seeing, what I'm showing you, I was more interested to look at the manipulation in the visual cortex and have a visual assay. And so that's why we did the visual task with the manipulation in the visual cortex. And one thing I can tell you is that there has been some literature in autistic patients as well, showing that they have different thresholds for detection, visual detection. The literature is a bit muddy, so depending on what kind, not the literature, probably the data itself, depending on what kind of autism, what kind of group, you can have increased perception or decreased the perception. So I cannot really directly compare, but visual changes in visual threshold has been reported, and these mice have some of the generic effects, such as marble bearing, nest building, and some of the social interaction tests as well.
Thank you.
Thank you.